Hello, everybody. Welcome. It's so great to see you all here. My name is Dion Rossiter. I am the Executive Director of Science at Cal, and I am so grateful to have you for our April lecture. We are taking the month of May off, as we normally do, and we will be back in June with 3 more lectures, so stay tuned. If you're not on our newsletter yet, please join because you will learn so much more about all of our Saturday morning lectures and the rest of our suite of lectures, which you'll hear about in just one second. Before I introduce our speaker, I'm going to do a quick land acknowledgement, if you don't mind. We recognize that Berkeley sits on the Huchun territory, the ancestral and unceded land of the Ohlone people, the successors of the historic and sovereign Verona Band of Alameda County. This land was and continues to be of great importance to the Ohlone tribe and other familial descendants of the Verona Band. Every member of the Berkeley community has and continues to benefit from the use and occupation of this land since the institution's founding in 1868. By offering this land acknowledgement, the Berkeley community not only recognizes the history of the land on which we stand, but also recognizes that the Ohlone people are alive and flourishing members of the Berkeley and broader Bay Area communities today. So thank you so much for allowing me to give that land acknowledgement. Science at Cal celebrates science. We bring the wonder and excitement of UC Berkeley research to the community. All of our events and programs are free and geared towards public audiences. So if this is your first event, welcome. It's so nice to have you. Raise your hand if this is your first time at a Science at Cal event. Yes, yes, I hope to see you at our events again soon. We work with tons of different partners across the Bay Area. We work with consulates and libraries. We're at First Fridays, farmers markets, community events. We're everywhere and everything in between. We, again, not just host lectures, we do cafes, we do exhibits, we do festivals, we're at Cal Day, and more, ... We'd like to thank Berkeley Community Media. You'll see there's cameras all around us, and these are folks who are recording for us today. So this will be recorded. It also will be online. It will be on YouTube. So we'll share this with you all who registered after the event and on our newsletter for those who couldn't make it. I'd like to thank the Science at Cal Advisory Council, who continues to support us, the UC Executive Vice Chancellor and Provost, who funds and supports Science at Cal, the Lawrence Hall of Science, who manages Science at Cal, and of course, last but definitely not least, our donors. Thank you so much if you donated today or if you donate in the future. We believe that science should be free and accessible for all. That's why we don't charge for any of our events and programs, but we can't do that and continue to do that without you. So if you are able to donate, please do. You can donate with Elise in the front or using this QR code. We have the QR code set up in the front as well too. Our next event is on April 21st. It is a great new future of plastics. This is our Midday Science Cafe where we feature UC Berkeley scientists alongside with Lawrence Berkeley National Lab scientists at noon every third Thursday of the month. So we hope to see you there. We're also going to be at the Bay Area Science Festival. We have 4 programs, maybe even 5. We have one that might sneak up on us. But our 4 programs we will definitely be at. One is at Discovery Days, which will be at Oracle Park on Sunday, April 24th. And that's between the hours of 11 to 4. We'll be, oh, you know what? I think I have slides for every single one of these. So that was us at Oracle Park. We also will be, we're supporting and partnering with the Unconscious Bias program. And this is a online virtual lecture on April 25th from 5 to 6. So please join us if you can. We will also be hosting a live virtual event in Spanish, and we have some really amazing 6 tours with scientists on campus, and it will have Q&A times after. And I just have to say, we're going to also thank our photographer and videographer, Vivi, right here, who, yes. So she's producing and directing and editing and putting them all together for us as a volunteer, and we're so incredibly grateful for her work and for taking photos and capturing our events. And Science Palooza, last but not least, I hope you will be able to join us or spread the word about our big event on the plaza of the Lawrence Hall of Science. It will all be open, it'll be free. We have 18 different departments who are going to be boothing, departments and student groups and partners who are going to be on the plaza, and it's going to be a great great big show up in the hills at the Lawrence Hall of Science. There is also, it's a $5 day at the Lawrence Hall of Science, so if you're a student or a faculty or staff, you get in for free. But you can also, if you're not, it's only $5. But please reserve your tickets in advance because they're doing limited entry. Woo! That was a lot, but you're not here to hear me talk about our future events. You're here for a current event, right? So I'm going to go ahead and introduce our amazing speaker today, Dr. Richmond Sarpong. He is a professor of chemistry. He's also the executive associate dean in the College of Chemistry, obviously at UC Berkeley, where he and his group specialize in synthetic organic chemistry. Richmond became interested in chemistry after seeing firsthand the effectiveness of the drug ivermectin in combating river blindness during his childhood in Ghana, West Africa. Richmond describes his influence and inspirations in a TEDx Berkeley talk in 2015 called The Face of Disease in Sub-Saharan Africa. I am going to let him talk about his undergraduate life, his life after undergraduate, and his current life now, probably, and his life in West Africa as well. So why don't we give a nice round of applause to Richmond Sarpong. Thank you.
Well, thank you very much, Dee, for a real warm welcome there, and thank you all for making the trek over to the Valley Life Sciences building on a Saturday morning. I appreciate you taking the time to be here today. And so what I'm planning to do today is tell you a little bit about not only the work that we have ongoing here at Cal, but also what influenced me to move in that direction. And so I am an organic chemist. And so you might ask the question, well, with being an organic chemist, what image does that really conjure, right, in terms of being an organic chemist? And of course, for I think all of us, when we think about the word organic, perhaps the first thing that comes to mind is organic food. And of course, living in Berkeley, organic food is something that I think all of us are very familiar with. Some of us might think about botanicals and cosmetics, for example, as being organic. And of course, there are, organic foods like fish and meats that might also come to mind. Perhaps what might not come to mind as immediately is pharmaceuticals and nutraceuticals, although for some of us, I believe, we might think of those as being organic as well. But in fact, when I think of the word organic, the first thing that comes to mind is people. And so here's a picture of me as a very young person. I was 5 years old in that picture. So I was born in Ghana in West Africa. And so here's a map of Africa. And I've sort of highlighted Ghana here. And that's because there's going to be a quiz after this where you will have to fill out the rest of the countries in Africa. Okay? I kid. All right, so here is another map of Africa with some of the countries filled in. This is actually a little bit of an older map, and so the true quiz is for you to figure out which countries have had name changes and which additional countries have been introduced since this map was put together. In any case, here's Ghana, and so I grew up in Ghana, and my parents were very much in the area of science and education. So my mother was an educator, a grade school educator, who in fact was my fourth grade teacher, and my father was a medical doctor. And as a medical doctor in Ghana, he was actually in charge of the upper region of the country. And so he was part of the government. And there was a coup d'etat in the country where the government was overthrown. And so that meant that my parents had to leave the country. And so we moved from Ghana, and we moved to Zambia, which is shown here. And after a few years in Zambia, we moved to Botswana. And so you might ask, why were we moving across from Zambia to Botswana, for instance? Okay? And that is because we were following disease. Okay? So let me clarify that a little bit more. And so here's a map of Botswana, and it's a lovely country. In the west of the country is a semi-arid area. It's a desert. It's called the Kalahari Desert. And for those that are not familiar with the Kalahari Desert, it's a very interesting and rich area for Botswana, which houses a group of people who are indigenous to that region called the San people. And so if you're not familiar with the San people, you may be familiar with a movie called The Gods Must Be Crazy. And so if you haven't seen that, perhaps, Netflix that tonight, okay? But it's an interesting story that is based on a Coke bottle, okay, if that tells you anything. But these are people that live very simply, and they subsist on the land, and so they're hunter-gatherers. And so juxtapose that with the rest of Botswana, which it turns out is one of the world's largest producers of diamonds. And so a lot of the De Beers diamonds, which you may be familiar with, which presumably come from South Africa, are actually mined in Botswana. And so, not surprisingly, mining is a big deal in Southern Africa. And so this blue line that is shown here actually represents a road that leads all the way to Johannesburg in South Africa and what are called the Kimberley mines, where a lot of mining for gold, for instance, happened. But unfortunately, what that meant was that a lot of people traveled all the way from Central Africa through Botswana to South Africa, and that also led to a radial spread of disease away from this road. And so, in fact, when I lived in Botswana, about 33%, okay, about a third of adults in sort of the in the age group of maybe 20 to 60 were in fact HIV positive. And this is part of the reason why my parents and my father in particular moved to Botswana, because he worked with the United Nations and the World Health Organization to help to try to curb this disease. And so disease was actually a big part of my life growing up. And not necessarily HIV/AIDS, but parasitic diseases like malaria, bilharzia, and elephantiasis. So as a 5-year-old, these diseases were all household names to me. And in fact, you were not a card-carrying Ghanaian unless you had contracted malaria at least twice, okay? And in fact, I did get it 3 times, so I could claim being Ghanaian, all right? But it wasn't necessarily these diseases per se that made an indelible impression on me. It was actually the world's response to the diseases like onchocerciasis. And so this is another parasitic disease that is caused by a black fly that bites the victim, and it's very generous, and so it leaves behind a parasite as a gift, okay? And so here's a parasite actually emanating from the antenna of this black fly. But that parasite ultimately causes blindness, okay? And that's because it travels to the eye membranes, and it eats away at the eye membranes. And so this can cause blindness. And so, as I mentioned, of course, it's a parasite that exists very much in the tropics, okay? And that's because its life cycle is along the banks of rivers, and it needs sort of temperate weather in order to thrive. But really what made an impression on me was this compound here, which was the world's response to this parasite. So this is a compound called ivermectin, and it's an example of a natural product. And that's a lot of what I'm going to be talking about today. And this is a compound that was introduced to address this disease. Now, ivermectin might sound familiar to you because more recently it's emerged as one of those compounds that people think might be important in addressing COVID-19. All right, and so it turns out that for those that are uninitiated, okay, the lines that are shown here and these hexagons and so forth, many of these lines represent carbon atoms. And these carbon atoms are linked to each other, and that is the basis for a lot of organic molecules. And so natural products are an example of organic molecules, and as noted by our famous journalism professor here at Berkeley, Michael Pollan, natural products are mostly carbon-based, right? And so, in many ways, carbon defines life's quantity. And so this compound, ivermectin, was very effective at addressing this disease, river blindness, that ultimately it led to a Nobel Prize in Physiology or Medicine, which is awarded in 2015 to Satoshi Omura from the Kitasato Institute in Japan, who actually first discovered this compound, and Bill Campbell, who was a scientist at a company called Merck Pharmaceuticals, who actually worked out the ability to derivatize this compound to address river blindness. And so as a 5-year-old, I learned about this story, and I was fascinated. And so I started looking for any literature that I could find that would give me more insight into this particular story. And my father had a copy of this book called the Merck Index, which contained lots of different molecules, and it talked about how they address different diseases. And he also had another book called the Physician's Desk Reference. And that really led to my interest in chemistry. I was essentially hooked as of that point. And so fast forward, I went to college at a small liberal arts college in Minnesota, and then went to graduate school on the East Coast, and then moved to the West Coast to do a postdoc at Caltech, and then started at Berkeley in 2004. And at Berkeley, my research group, which is all the way close to the Greek Theater over in Latimer Hall, focuses on natural products. And so I wanted to tell you a little bit more about natural products and why I'm so excited about them. And so the first question you might ask, and I sort of gave this away a little bit at the onset, is where did they come from? And natural products, by and large, are isolated from both marine and terrestrial environments. And so here are some organisms that, in fact, are the basis for different natural products. And so from the skin of frogs of this type, you can isolate toxins, which are in fact used to tip the arrows, okay, of different indigenous tribes in the South Americas. And this compound here produces a compound, a natural product called wolfsbane, where if you've read Harry Potter, in fact, Snape talks about wolfsbane, okay? All right, very good. And so they're isolated from all sorts of different sources. And the reason why these organisms produce these natural products is that over the billions of years of their existence, they have evolved to use natural products which are produced by these organisms for either communication or, in many cases, to ward off predators. Okay, and so for example, by displaying these very bright colors, it tells a predator, I have these very noxious natural products, and so stay away, okay? And as we will learn, these natural products can come from very, very interesting, cool sources, like what I show here, but they can also come from pretty boring places like golf courses, okay? And so it turns out ivermectin was actually isolated from a golf course. And so Satoshi Omura was out for a round of golf, found a very interesting soil sample, isolated that, and cultured it, and isolated this compound. Okay, so why am I so excited and standing in front of you here, frothing at the mouth with excitement, telling you about natural products, okay? And the reason I'm excited about them is because they have a very clear connection to disease, all right? And so don't pay too much attention to the structures, okay? What I want you to pay attention to are the different diseases that are listed underneath these names. It turns out the natural products, by virtue of their diversity, have a very interesting function. And that function can be applied to addressing different human ailments, such as the ones that I indicate here. All right, and so this is obviously somewhat of a subset of the big, full picture. And so people have done different analyses. And one of those analyses that's been done showcases how natural products actually overlap pretty significantly with different medicines. And so here in green, I show different natural products, and in blue, I show the sort of landscape, if you will, of where you have different approved medicines. Okay? And it turns out that there's actually a pretty large overlap between approved medicines and their landscape and the landscape of natural products. And so, in fact, even with the emergence of very, very interesting biologics to address disease based on enzymes and so forth, and natural products still account for about 50% as the basis for medicines that we all use. Okay, and so today I'm going to walk you through a little bit about the different natural products and how they connect to different diseases. Right, so one reason why we should make natural products clearly is because they could serve as the basis for identifying medicines to address disease. But the other reason, and why else we should make natural products, is maybe much more academic. And so I'll give you a sense for that. Okay, because I look at natural products and the types of structures that I showed to you, and I see them as architectural marvels. All right, and so here are some examples of architectural marvels, right? And so a little quiz here. So that's a geodesic dome, and those were popularized by a person called Buckminster Fuller. And so that then became the basis for Buckminster fullerene, which is an allotrope of carbon and was also, in fact, a basis for a Nobel Prize. And so here's Buckminster Fuller in front of one of these geodesic domes. You will recognize the Guggenheim Museum in Bilbao, and this building you might recognize as well, right? That's the Millennium Tower in San Francisco. So, in many ways, natural products are viewed as architectural wonders, and just like architects and as people that build buildings, we have to learn how to come up with blueprints for putting them together, and then we have to go through the process of identifying the tools and the strategy that we need to build them. And that process leads to technological advances in terms of the types of tools that we're inventing and the types of strategies that we use to build these buildings. But as you know, every once in a while, there's a mistake, okay? So you try to put together this building, and, well, it starts to careen to one side, as is true for the Leaning Tower of Pisa, and they become tourist attractions, right? And as you may know, that is also true for the Millennium Tower, which is now leaning, okay? But in any case, they can be happy accidents where this can become a tourist attraction. And so, in my opinion, the why else we should make natural products is because we view them as sort of a goal that we must reach. For example, getting to scale amounting to get to the summit, right? And so we have to identify a path in order to be able to do that. We have to gather the right tools to enable us to do that, and of course, have the right strategy for how we're going to get to the top of the mountain. And along the way, if we veer from a traveled or well-traveled path, we might in fact come up with new discoveries and happy accidents. And so the way in which I view science is very much through the lens of really thinking globally, okay? And that is pretty much based on my childhood. And so here I'm showing the sort of sustainable goals as laid out by the United Nations. And so I encourage all of you to sort of look at these and think about perhaps how we can all contribute. And one area where I thought we could make a contribution was in thinking about really improving the well-being of society. And so with that said, when I came to Berkeley, one of the first areas that we worked in was to think about diseases that are found in tropical areas. And in particular, we became interested in thinking about malaria and ways in which we could help address malaria. And so, like river blindness, a lot of the cases in malaria are in Sub-Saharan Africa, and it turns out that there are many medicines that have been introduced to try to address malaria. And one of the most effective medicines, and in fact one of the last most effective medicines, because there's been a lot of resistance to the existing medicines, is this compound called Artemisinin. And so, in fact, it's effective, but the challenge is that it's very expensive. And so if you're going to have people in areas of the world who are not as financially able afford this, the challenge was to produce this for about 25 cents a dose. And so when I arrived at Berkeley, we started to work in collaboration with another group here at Berkeley, and this is really, to me, the power of Berkeley, is collaboration. Another group, which was a group based in chemical and biological engineering, run by Jay Kiesling here at Berkeley, and what they had shown is that if you take yeast cells, so if you take yeast, you can actually engineer the yeast cells, and you can arrest their synthetic machinery for making different molecules, and you can essentially coax them to carry out different transformations, as I lay out here. And we corroborated these transformations in the chemical laboratory. And so using yeast cells now, you can essentially do a fermentation to produce a lot of this material. And that then sets the stage to make these molecules at a low cost, arriving at that goal of about 25 cents a dose. And so Jay Kiesling, who is shown here, has gone on to scale up this process and has started an institute for this, as well as a company which now uses this technology to produce artemisinin on a large scale affordably for the world. All right, another area that we started to think about was to think about the alleviation of pain. And as you may be familiar, opioids are a very important part of addressing pain. And in particular, morphine, the structure of which I show here, is a very important opioid for addressing pain. Now, as I think has emerged over the last decade, there's real challenges with using morphine, and so there are alternatives that have been proposed. So, for example, non-steroidal anti-inflammatories, such as the ones that I show here. So these, by the way, are not natural products per se, whereas morphine is a natural product, but they're effective. But often they're effective for a short period of time, and they have their own challenges in terms of association, in some instances, to heart disease and liver disease and so forth. And so the opioids are very, very effective, but part of the challenge is that the opioids, like morphine, are very closely related to other compounds like that, which, of course, has serious addictive side effects, and this is a big problem. And so when we started to think about a way in which we could contribute, we decided to first learn about the basis for pain. And so I'm going to share what I learned with you. And so what I learned is that pain is caused at the molecular level by events that are happening at a receptor. And that receptor is a protein, and so this is a structural representation for a protein, a cartoon-like representation. It's a little bit complicated here, okay? But the point is that when you have this protein, and I've shown a cartoon of it here, okay? If it opens, and it opens to unveil this sort of pore in the protein, sodium can travel through that pore and into, for example, your nerve cell. And that causes a change in the potential in your nerve cell, almost like what leads to electricity, right? And so that causes an action potential, and this then essentially travels along the nerve cell, and you perceive it as pain. And so if you were bitten by a scorpion or a snake, what happens is that they produce toxins that bind to these ion channels, and they keep this pore open. And so lots of sodium travels in, and that causes a lot of pain. And in fact, if a lot of sodium keeps traveling in, it will saturate the signal, and you will go into rigor mortis and eventually die, right? So that's what the toxins will do. If you go, on the other hand, to the dentist can deliver a local anesthetic like lidocaine, And that, as represented here, can bind in the pore of the channel and prevent the sodium from traveling into that nerve cell, and so that blocks the pain sensation. And so what I learned is that, in fact, there are ways in which you can control pain at the molecular level. And so we were very excited when we learned about these types of natural products, which are in fact used in traditional Chinese medicine to address diseases like cardiac arrhythmias and atherosclerosis. And what we learned is that these molecules are also effective at interacting with those channels that I showed. But we also learned that there were about 10 different channels. Okay, so there were some of them that are associated with things like epilepsy, others that are, of course, associated with cardiac processes, and hence they're used for cardiac arrhythmias. And there were 3 of them that were associated with pain. And so if you wanted to block the pain sensation without causing migraines or to avoid causing cardiac challenges, then you had to selectively interact with these particular variants of these particular ion channels. What was also exciting is that it turns out that there are actually people that have a mutation, and the mutation is such that they don't express these particular ion channels. And so those people are impervious to pain. They don't feel pain. It's pretty amazing. And so you might have been, party to, a circus or something of the sort where you've seen people do this, and it's real that some people don't actually feel pain. But then there are other people that overexpress these particular channels, and those people feel a lot of pain. In fact, just touching them causes excruciating pain. But what all of that means is that, in fact, we have a human model for this to show that if you can block this, you can, in fact, on a temporary basis, that is, you can, in fact, relieve pain. And so we asked the question whether we can start to achieve specificity in blocking only these channels and not the others. And so the first step to do that is to make particular variants of these molecules that I showed here, which would enable us to interact differently with these channels and enable us to gain selectivity in blocking one of the channels over the other. And so we've done that. So we've done the first step, which is to start to make different variants of these natural products to start to think about interacting differently with the different ion channels. All right. And so, in fact, that's what my group mostly does, is we work on making these different molecules. So, how do we make these different molecules? That's really part of the story that I want to tell you today. And it's very sophisticated, okay? It turns out we take the IKEA approach, okay? All right. And so the way we make molecules is very much like how IKEA furniture comes to you. And so you obviously want to buy, for example, a chair from IKEA, and what you're going to get are the pieces of the chair. And so this is the type of exercise that we carry out in our group, where we essentially come up with an exploded diagram of those complex structures that I showed to you, and we essentially take them apart in the reverse direction as an exercise for how we would then put them together in the forward direction. And so this is a process that we call it break it to make it. So to make it, you first have to break it apart, all right? And so this process, as you can imagine, there are many different ways in which you could imagine breaking this chair apart. And so you can come up with different permutations of how you carry that out. And sometimes that's overwhelming, but you get trained in how to do this. And more recently, we've, like many in the type of science that we do, have recognized that, in fact, to gain a handle on all the different permutations, a way to do that, perhaps, is to computerize this whole process, okay? And so can we start to computerize the different permutations of different ways of breaking apart the thing that you would like to make? And this becomes a little bit esoteric, so I'm not going to get into the details of it. But we start to look at these molecules, and we start to treat them as molecular graphs. Okay? And as we walk the different graphs, we can determine how we can achieve a way to simplify the structure as we take it apart to its constituent parts. Okay? So this type of process one can carry out using computations, and this is something that is exciting and emerging. And it's very much connected to another exciting, emerging buzzword that you've probably heard, this idea of artificial intelligence and machine learning. And so this is something that as a field in organic chemistry, we are starting to utilize in terms of how we think about making molecules, where you can essentially learn from all the existing data for how people have made molecules and now start to make connections between the different pathways that people have identified for making molecules to guide how you might make a molecule of interest to you. All right, so again, if I return to this particular graph that illustration that I showed early on, climate change is another area that I think is on all our minds. And when we think about climate change, I would say, by and large, I think for a lot of us, it begins by thinking about oil and drilling for oil, which has obviously led to a lot of the challenges that we all think about. It so happens that drilling for oil and a byproduct of that industry is, in fact, the starting point for a lot of organic chemistry. And so these are the types of byproduct molecules that come as a result of drilling for oil, and these are the materials that are often used as a starting point to make a lot of different things, be it new medicines or materials, like different types of plastics or other types of polymers that are the basis for our clothes and materials that we all use. And so it's an important part of our world, but obviously it comes along with challenges. And so as we all head towards 2050 and try to wean ourselves from fossil fuels and their byproducts, we in my lab have become interested in thinking about sustainable starting materials for synthesis. And it so happens that in agriculture, there are different waste products that emerge from the agricultural industry and different byproducts. And some of those are great starting points for synthesis as well. For example, this compound that I show here, which can be obtained from either caraway or spearmint. And in fact, if you take orange peels, you can start to isolate some of these molecules from there as well. And it's a little bit of an optical illusion here, okay? But it turns out these two compounds are what are called enantiomers. So they're mirror images of each other, just like your hands are mirror images of each other. And mirror images are a very important part of life because it turns out that each of those mirror images will interact differently with receptors in your body. And so you want to be specific about which mirror image you're using to make, for example, a drug. And so we became interested in using this as the basis to make this compound, which is another natural product. It was a natural product which was isolated from the back of a snow crab off of the coast of Okinawa in Japan. And so somebody was out collecting snow crabs for dinner, okay, and happened to notice a fungus growing on the back of the snow crab and said, I wonder what type of natural product that contains, okay? So the next time you go to make your dinner, look out for the different fungi that might be on your food. Okay, in any case, they cultured that fungus and they isolated this compound and then they tested it for activity. And it turns out that it emerged as a lipid mediator of leukocyte function. Okay, that's a mouthful. But bottom line is that that connects a lot to inflammation. And so as you may know, a lot of diseases are connected to inflammation. And examples I show here. And so people thought maybe that could be the basis for trying to address some of these diseases that are associated with inflammation. But again, we wanted to understand exactly what aspects of this compound make it function as an anti-inflammatory and what types of diseases it might enable us to address. And so we decided to make not only that initial compound, but many other related compounds, as I show here. And in synthetic chemistry, we mark the level of efficiency based on how many steps it takes to make it from commercial materials, and also what the yield for these materials would be. And so the bottom line is that we could make these materials. And so once we made them, then we wanted to learn about their function. And as I mentioned, these compounds were known to have anti-inflammatory properties, and so we decided to study them, particularly for cancer. And so in a collaboration that we carried out, we took some cancer cells, okay, and one way to kill cancer cells, as you may know, is to attack them with radiation, right? And so we can blast these cancer cells with gamma rays, and that basically leads ultimately to the cancer cells dying. And the way in which they die is that they lyse, so they essentially explode, okay? And when they explode, all the inerts are released, and it turns out that some of those inerts contain what are called growth factors. And so if you take those cancer cells that you irradiated and you layer on top of them other cancer cells, which are now tagged with fluorescence using what is called green fluorescence protein, you can in fact use fluorescence to watch how quickly those fluorescent cancer cells can grow. And it so happens that if you just have those fluorescent cancer cells and you look to see how easily they grow, this is represented here in this bar graph, they don't grow that fast, okay? The higher this is, it means the more fluorescence, it means they're growing fast. They don't grow that fast. However, if you layer those fluorescent cells on cancer cells that have been irradiated, they grow really fast, okay? And if you take one of the best known ways to reduce their rate of growth as a result of the released growth factors, it slows down the growth here. And it turns out that the compounds that we made, those also slow the growth, as shown here. And in fact, some of them are pretty effective at slowing the growth of these cancer cells, the regrowth of the cancer cells. And so this is not a cure for cancer, but what it tells you is that these molecules have the potential to slow down the regrowth of cancer cells after you've irradiated to kill the majority of the cancer cells. Okay, this is a process which is called tumor repopulation. In fact, it's a very common way in which cancer cells can hijack a lot of natural ways to essentially regrow. And it's a very common mechanism, in fact, is the same thing that happens in wound healing. So for example, if you get a cut, there's inflammation in that area, and that triggers this process of regrowth of your cells. It's a repair mechanism, and the cancer cells have essentially hijacked this repair mechanism. And so this process can somehow start to slow down that repair mechanism for the cancer cells. Okay, so I've cheated a little bit, okay, which is that I've mostly told you about natural products. So I haven't told you the whole truth, all right? And natural products, as I represented early on, are over here in green. But it turns out that for approved medicines on the market, they're not all based on natural products. For example, the orange dots that are represented here on that landscape, those also overlap with approved drugs. And so we wanted to learn a little bit about those as well and how they might play a role in approved drugs. And so we decided to study what those types of orange circles represented, and they represent molecules that kind of look like this, okay? And those are, in fact, the basis for a lot of drugs as well, a lot of medicines. One that particularly caught our eye was this one, and that, as you can see, and here I'm going to have you think a little bit about structure, you can see that it's related to this structure here. So there's a 4-membered ring, like a square, and this is penicillin. All right, so penicillin and related compounds are, in fact, the basis for a lot of medicines. And so penicillin is a natural product, and it's probably one of the most famous natural products. And again, it's a natural product that is isolated from a fungus. And so here is the fungus producer of penicillin. And so that looks a very cool picture, right? But this is actually the original culturing of penicillin. And you can see the culture here, and there's essentially empty space away from that culture, and other things grow. And so basically what that tells you is that it's able to kill anything that's around it. There are other bacteria, all right? And so, of course, it's a very potent antibacterial compound, which was discovered by Alexander Fleming, who was a scientist at Oxford. And once he discovered it, there was a lot of excitement about it, because prior to that, once you got an infection, you sort of, just said your prayers and hoped for the best, right? But after he discovered this, it became very important in terms of how to address disease. And the very, very first patient for penicillin was this person here called Albert Alexander. And Albert Alexander was a police officer who is believed to have been injured by a rose bush, okay? So he got a huge gash from the thorn of a rose bush, and that got infected. That's the lore. The reality is probably somewhere else. I mean, some people believe that he actually was he was in the war, and there was a blast, and he got injured that way. But he got a pretty bad infection. And so once Fleming had discovered penicillin, they decided to try that on Alexander here. And it turns out that it actually worked very well. It worked really well for about 1 week. He was getting better. And then, unfortunately, he got worse and he died, okay? But that was because they used impure penicillin at that time. They weren't, sure about exactly how to purify these things. And they also exhausted the world's supply of penicillin at that time that Alexander Fleming had produced. And so that's why that happened. And as it turns out, they recognized the success, and these were the limitations. And so that actually sparked a huge collaboration to produce a lot of pure penicillin, which was very important in the war effort, as you may know, in the 1940s. And ultimately, over time since then, it's become less effective because of resistance that has arisen out of a result of using it too much, right? And it turns out that even though resistance to penicillin has emerged, you would think we would work really hard on coming up with other antibiotics, but the business of antibiotics is actually not that lucrative, okay? Because you want to constantly produce new medicines, and if you come up with one that's really, really, really good, then it just means that there isn't the business of making new ones. And resistance can arise for these types of things. So I wanted to end here and just sort of share this with you. So a lot of the antibiotics that we have, which are, by the way, based on natural products, because that is nature's way of dealing with other organisms, they're all very old molecules. They've been around for a long time, since the 40s, 50s, and 60s. So for example, the commercial derivative of penicillin was introduced a long time ago in 1964, based on this natural product. It's effective against gram-positive bacteria. So it turns out there are 2 different types of bacteria. Those that are gram-positive, it turns out those are the easy ones to kill. Okay, and then there's gram-negative bacteria, which are harder to kill. And these molecules, by and large, are really effective for the gram-positive, easy-to-kill bacteria, not as effective for the hard-to-kill bacteria. But through chemical synthesis, which is the type of exercise that my laboratory does, you can, in fact, start to change these molecules and introduce different aspects to these molecules that actually can make them much more effective at killing not only the easy-to-kill bacteria, but also the hard-to-kill bacteria that is said to have broad-spectrum activity. And so we are very hard at work thinking about ways in which we can start to contribute to this overall effort here at Berkeley. And so, for example, here is ongoing work in our laboratory where we're interested in simply using the sun's energy to guide how we do chemistry. And so we can take compounds like this that are easy to access, and we can start to make compounds like that. And you can see that these are somewhat closely related to these types of molecules. And so we are interested in derivatizing these and preparing different derivatives that might enable us to continue to tackle this problem of antibiotic resistance, which is a real problem. I realize that COVID and vaccines are very much on our mind, but believe you me, antibiotic resistance is a war that we'll continue to fight. All right, so I'll end there and show you this picture. This is Satoshimura, who discovered ivermectin. And here he is in Ouagadougou in Burkina Faso in West Africa. I could have been any one of these children in 1980, okay? And of course, here he is again, as I mentioned, won the Nobel Prize for that work. This picture, in fact, does contain me in that picture. And so part of your quiz today is to find me in this picture, okay? I'm actually back here, okay? So I come from a majority black country. You know, I've been very fortunate and humbled to have had an opportunity to visit many parts of the world. And so here's a picture of Satoshimura and I, along with another professor that he works with in Japan in 2018. And so his group and our group has had a very fruitful exchange of different scientists that have come from his laboratory to work here at Berkeley in our laboratory. And so while it's difficult to find me in this picture, it's probably easier to find me in this one. Okay, so there I am. So this is my research group at UC Berkeley, now taken about a year or so ago when we were allowed to take off masks for a brief period of time. And it's a very diverse group of individuals that I'm very, very fortunate and proud to work with here at Berkeley. And so again, the diversity is something that we really celebrate in the group, despite the fact that we're all wearing uniforms here, okay? But they've taught me a lot, not only about chemistry and life, but also about what it takes to be a great coworker, which is represented here in this word cloud, okay? And I think these are sort of lessons that all of us should take away in terms of how we interact with each other. And so I really encourage you to keep these types of virtues in mind in how we interact with each other, especially in these trying times. So I will stop there for today and thank Dee and Science at Cal once again for the invitation to be here. Thank you all for your attention, and it's a pleasure to see that at 11 a.m. on a Saturday morning in post-COVID era, people still come together to celebrate science, and this is a real testament to the power of science, not only in this room, but at Cal and throughout the world. So thank you again. Much appreciated.
As normal, we'll do some Q&A. So go ahead and raise your hand, and Dr. Sarpong will repeat the question for everyone to hear. Before I do that, because it's always a little weird interrupting again to do another round, I'm just going to say thank you to him, and I'm going to provide, Elise and I would like to give him a Science at Cal shirt.
Well, thank you. Much appreciated.
For you to wear proudly while you celebrate science.
I will. Thank you.
If you want a different size, just let us know. But all right, so I'll let Dr. Sarpong run his own Q&A. So if you guys want to go ahead and ask questions, please do.
Right, and it doesn't have to be a scientific question. Feel free to ask me anything, since I opened with the childhood picture. Yes?
There is a tendency in biotechnology to design drug molecules, anticipating what to happen with them and how to produce them and how to store them. In other words, it's the idea of investing a lot of efforts in the initial stage of drug discovery in order for the drug to perform in all aspects of what we can think. And my question is there, in your view, any way to embed in the molecules that you are designing to address various problems in the gut space to avoid drug resistance, to avoid being pumped out from cancer cells? In other words, can we design the structure of the molecule so as to avoid the smartness of the bacteria to, say bacteria, we are talking about antibiotic resistance, to actually chew it very fast and avoid the use of the drug over a short period of time?
Well, this is an incredibly deep question, so I'll try to repeat it. The gist of the question is there a way to design into molecules a way to avoid resistance? And in particular, for a lot of molecules, a big part of the resistance mechanism is that the organisms to which the molecule is supposed to affect will pump the molecule actively out of that organism through different what are called efflux pumps. And so is there a way to design molecules to avoid this process? Well, a lot of times when molecules are taken into a cell, there are multiple mechanisms. They can be taken in actively or passively. And the process of being pumped out of the cell, it's often an active process, which uses up ATP and energy and so forth. And so you're fighting often a process of bringing in molecules through endocytosis or some other passive process with this active process of pumping it out. And so there are several things that have emerged in terms of ways to design molecules to favor actually this process of going to the cell. And so you can create what are termed sort of charged balance scenarios. So for example, in some cells, you might have a case that they're much more ionic or anionic, for example, or cationic. And so if you create a molecule of the opposite charge, it might have the ability to reside more in the cell. But that's a very general statement. Or in some cases, you can create a molecule where it arrives at its target, it causes the change that is supposed to happen, and then essentially falls apart, so that it's not necessarily the whole molecule that's being pumped out actively, but that it actually carries out its activity and falls apart. Or it could be that you design molecules which are what are termed catalytic, meaning that the same molecule can cause an effect that is maybe 1,000 times or 10,000 times where you would start. And so you'd need then a very small amount of the molecule to reach its target to cause, to have an effect. And so recently there's been a lot of interest in for example, a recent example is this idea of what are called PROTACs, which are essentially take a molecule and attach it to a protein, and this makes it into a cell. And if, for example, you're trying to get rid of a particular protein, once it binds to that protein, it can recruit a way to tag that protein for destruction. And so the same molecule can be used to destroy many, many copies of the protein. And so there are many different what are called modalities for addressing disease with small molecules that are emerging, which go beyond just using the small molecule by itself to for example, affect or inhibit or enhance the function of a particular protein. So again, this is maybe a little bit outside of my immediate wheelhouse, but I guarantee you that these are important questions that you're asking, and there's a lot of scientists that are looking at different ways to try to tackle it. But it's an important issue, especially when it comes to small molecules, this idea that, you have this active efflux out of the cells and out of the organisms. So all I provided was maybe a bit of an overview of different ways that people are looking to address this. Yeah. Great question. Yes?
Are there any situations, in your opinion, where it'd be advantageous to go the synthetic route rather than trying to find a natural product?
That's a great question, something that I probably did not address very well. So are there any scenarios where it's more advantageous to make something synthetically versus go after the natural product? And the truth of the matter is that if you take a natural product and you look at it at a molecular level, it's indistinguishable from another molecule that you made synthetically. Okay, so they're the same thing. So just by virtue of being a natural compound does not mean that it doesn't have the same potential challenges. Okay, so I think often when we say synthetic, we associate that with something negative, and it might be toxic and so forth. As I showed to you, even natural products that are produced from nature can be toxic. In fact, they're used as poisons and so forth. And so the advantage in many cases of making these molecules in a synthetic laboratory is that when you go out in nature, only small amounts of these natural products are often produced. And so you'll be destroying whole ecosystems by harvesting from a natural source a particular natural product. There's some famous cases. So for example, there's an anti-cancer drug called Taxol. And this is a very important anti-cancer drug, also known by the name of paclitaxel. And this was initially isolated from the bark of a tree called the Pacific yew tree. But when they harvested the bark of the tree, they essentially killed the tree. And from that bark, you can only isolate very, very small amounts. And so people decided to actually make it in the laboratory. And it's just as effective, and that now avoids this sort of ecological disaster of killing a lot of plant matter. As I mentioned, several of the natural products that are really of interest for disease are isolated from the marine environment. And so you will be destroying lots of marine ecosystems by harvesting these marine organisms in large quantities to address the supply needs that we would have as a community. So I would say part of the advantage, if you will, of preparing these molecules synthetically is that you can address a supply problem. The other advantage is that often through a chemical transformations, we can target almost with like laser-like precision at an atomic level different parts of the molecule, because often while the whole molecule might be advantageous in terms of addressing disease, it might have some side effects, right? And so you can program in, by virtue of how you manipulate the molecule, ways to enhance the positive effects and reduce the side effects. And so you can make derivatives of the natural molecule to enhance the positive effects. And so that is done through sort of synthetic manipulation. By the way, I think that those, often when we think synthetic, we're thinking of smokestacks and some, industrial complex process. But in fact, you can use natural organisms to also carry out these chemical transformations. So I mentioned yeast cells, for instance, that are being used, but you can also use other natural enzyme constructs to make these chemical transformations as well. So I think that the line is pretty blurred between molecules that would arise from a synthetic source or from a natural source. You can have good and bad from either source. Yeah, but great question. Yes?
This is kind of along the same lines. The natural products seem to have kind of two chief problems. You need to find a natural product, and you need to kind of know its influence in the application that you want to go for. I think both of those steps are
Yeah, this is a great question. So the question is, are there more direct ways to, if you will, design in an intelligent way molecules to address disease by looking at protein structures? So as I think a lot of people appreciate, a lot of diseases can arise as a result of, the aberrant function of different proteins. And so if you can, in essence, introduce a molecule that either reduces a protein that is overactive or enhances the activity of a protein that is not as active in a particular disease, you have the opportunity to address that particular ailment. And so absolutely. So what I told you here is just a very thin slice, which is from our vantage point. But with, of course, the Human Genome Project and everything that emerged out of that, there is now a much deeper understanding at the protein level, at the DNA level, of how one can address disease. At Berkeley, of course, you can't talk about science without talking about Jennifer Doudna and CRISPR-Cas9 and the ability to start to address how you arrive at proteins by, in fact, changes to the DNA prior to transcribing that and arriving at the protein. So absolutely. And so, in fact, there are many, many pharmaceutical companies, for instance, that look at protein structure. And often when you look at protein structure, you're looking at it in a static state, meaning you look at a crystal structure, or you can use electron microscopy, cryo-electron microscopy, which is also a big thing here at Berkeley, to look at those structures. And on the basis of being able to visualize those structures, you can start to identify pockets where molecules might be able to bind to either inhibit or enhance the activity. So if you bind to the active site of that particular protein, you can inhibit its activity. You bind to some allosteric site, you can cause a conformational change that makes it much more active. And so absolutely, there are companies that really just look at that and then design a molecule which would interact with either that active site or that allosteric site. And this is definitely the basis for also addressing disease. But it's just that natural products have had such a long history of being, just naturally evolved over billions of years to be perfectly tuned to interacting with these different proteins. And so it just provides a starting point, as opposed to sort of approaching this from a purely, scattershot, random starting place. You can hone in on perhaps places where people over billions of years have had success in evolving molecules to interact with those proteins. But it's just one approach. Yeah. I think you had a follow-up, or?
Well, I just was going to say, if the protein structure was very important for that, it sounds like the protein also responds kind of structurally to whatever stimulus you provide, in some cases at least. So like you're saying, kind of static picture is maybe not enough to
Right.
fully understand
Right.
the picture.
Absolutely. So the static picture is not enough. And so there's lots of people that work in protein dynamics to really understand how the protein might change a particular structure depending on what environment that protein is in. And so there's lots of computational models to sort of look at that as well. Yes, there's a question in the back.
Are there drugs in the market or in the pipeline that's designed or as a result of the artificial intelligence effort that you described earlier?
Are there drugs that have been designed or in the pipeline based on the artificial intelligence efforts that are ongoing? I will tell you that there are many companies that have sprung up in the last 10 years based on exactly this principle, that you can capture all the data of existing molecules, right? So it's very difficult for one individual to have command of what, let's say, 20,000 different molecules that have emerged as medicines, what features of those molecules are most important for why they're medicines, right? And so if you can look at all those different features and try to understand what aspects of those features lead to medicines, you could, in principle, design from scratch, ab initio, a new medicine. And so there are many companies that are emerging to do exactly that. I can't, off the top of my head, pinpoint a particular molecule that is, or drug that has arisen as a result of that. But I can tell you that often when you're developing a drug, that process of first identifying the compound that's active, and then going through all the different clinical studies to understand the side effects, how bioavailable it is, and all the other pharmacokinetics that surround that, that process takes about 15 years. Okay? And so if these companies sprung up, let's say, 5, 10 years ago, maybe in the next 5 to 10 years, we'll start to see these medicines emerge. So that's what I can tell you, but this is a very important part of harnessing all the data that we have, because I think there's been sort of an explosion in the ability to generate new data, and what's been lacking is the ability to harness and learn from that data. And so these companies are emerging to do that. And then obviously the proof is in the pudding, where once you identify a compound, you have to then go make it, test it, and see if it realizes its potential. So this is definitely ongoing. Yes?
So I'm looking at all these human characteristics that you're talking about here, and you were talking about trying to get inexpensive malaria drugs, and it made me think of this project my daughter was on dealing with sort of interaction between malaria and pregnancy in Malawi. And the problem they had there was the drugs were basically free, but these young women that would lose their first baby because they had a negative interaction between the malaria parasite and the fetus, they couldn't get to where they were getting the drugs that were free. And one of the problems was that they were required to get a letter from the village chief to go get the medicine. And the village chiefs would then charge for those letters. And so it's like a human thing. And so I guess I'm trying to tie into your growing up in Ghana and living in Zambia, that you've got all these human factors, and I wonder if you've been back to those areas and thought about the science that you've done here at Cal and talked to people back there. And it's just a very different environment. And if you see any hope for improving the environments there, getting more science into them.
Yeah, well, the question and the commentary that you just communicated, and I hope a lot of people here heard it, was really about the sort of human challenge of actually even gaining access to particular medicines that are freely available. And I would argue that while this was true in Malawi, where your daughter was working, it's in fact true in many parts of the world, not only Sub-Saharan Africa. I've heard, sort of stories from the Middle East, as you would imagine, where, again, having access as a woman to a lot of resources becomes a big challenge as well. Southeast Asia in some places, and so these are real human problems. And so, one of the reasons I got into chemistry is obviously for the love of the chemistry, but also because I realized that this work was done by humans. And I'm very interested in those stories of human beings making a positive difference in the world. And obviously one way in which you can do that is through chemistry, but I would hope that all of us as people would recognize that there are other challenges like what you mentioned. And so it frustrates me and it saddens me to hear those stories and to know that they still exist. But what in my little part of the world I can do is to bring awareness to these things, and it clearly sounds like you're bringing awareness to this as well. And it's a shame that we continue to have these challenges where somebody always looks for an advantage in a situation where others are suffering. And clearly, even today, we see that happening all around us in the world. So I don't have an answer to your question, but I'm certainly aware of this, and the way in which I try to be helpful is to shed some light on some of the positive outcomes that can arise, coming from modest beginnings, and hopefully, there are opportunities to shed light on exactly the types of problems that you're pointing out, and perhaps bring justice to the village chief that is extorting people to write these letters. It's amazing when I hear these stories. I don't really have an answer to your question, but I'm glad you're bringing awareness to this, and I hope that authorities are aware of this. But you're right. I mean, we perhaps are in a very privileged environment where, you know, the challenge is to actually identify the medicine to address a particular disease, whereas in other areas, I mean, you know, malaria I talked about in Artemisinin, which is amazing. For a lot of people, just even having a mosquito net to cover their bedding and prevent, you know, the bite from the Anopheles mosquito in the first place is a big challenge, right? Or having clean drinking water is a big challenge. And so there are these sort of basic human, you know, needs that we still have as challenges around the world that we hope will be addressed, not by others, but by all of us being aware and speaking up for the rights of others. So I appreciate and applaud the work that your daughter is doing, and thank you for your comment.
I wanted to ask you about what's the most rewarding part of your job is, and what's interesting to you, and also what's challenging about what you do.
Yeah, so the most rewarding part of my job is really my interactions with my coworkers, the people that I work with. These are often young people between the ages of 18 and 32 or so that are really excited about science and challenge me every day in terms of my preconceived notions about how we should do the science, and teach me something every day. I love being in a university environment because surrounded by young people that are very enthusiastic, very passionate. And to me, it's really great when I learn something new from them, okay? Because I always do tell my coworkers that, you know, we're in trouble if the extent of our knowledge is what I know, right? I really appreciate that people bring new things to the table, so I really love those interactions. Perhaps the biggest challenge that I've had, I would say, is in what I do is really has to do with sort of the magnification, if you will, of a lot of mental health issues that have arisen, you know, as a result of, obviously COVID has magnified that, but I think that there's been a rise in sort of the challenges that many people face. And so it's you know, been a challenge to try to help where I can with people that I interact with that are going through these challenges. It's very real. So that's what I would say. Thank you.
Two of the examples of like where the molecule, I guess the organisms or natural products are from, were like really random. You said one was from a golf course and one was from like the fungus on a snowcrab. So I was wondering if there's like a more directed way of deciding like which microorganisms to look at, because otherwise it seems like you're looking for a needle in a haystack. It's like really hard to find the right organism for the right molecule.
Yeah, it's a very good question. So is there a much more intentional way to go about finding natural products that are effective as medicines, is really your question. And so, you know, you'd be surprised, but in fact, this needle in the haystack thing was very much the way in which things were done a long time ago. And so many people, for example, have heard of Richard Nixon, and often in a very negative sort of connotations, but one of the things that he introduced was this war on cancer. And so, you know, the NIH, the National Institute of Health budget sort of doubled, and there was a lot of, you know, investment in addressing disease. And that actually led to increases in the budget for essentially scouring the earth to find different molecules. And initially it was done, as you said, in a very haphazard way. People would, for example, talk to shaman and fetish priests in different communities because those were, you know, concoctions that they were using that actually had in them different natural products that were effective at addressing different diseases. And so those were isolated in that way. But more recently, people are essentially looking at genetic maps of different areas And on the basis of looking at data and learning, machine learning from that data, you can start to predict different genetic constructs that might actually connect to different molecules that would be the basis for medicines. And so there's sort of a meta study of genetics of different organisms that might actually represent sources of medicines. Because one of the other things is that often, for example, with plants, they will only produce a particular natural product only when they are stressed. So for example, they have a lot of insects attacking them, then they will go into a state where they will produce those active ingredients, active molecules. And so it's not always. And so they might still contain the sort of genetic code to produce that, but it only is activated, if you will, when they are stressed. And so ideally, what you would like is to sort of map out their genetic code and recognize, even if they're not produced in a stressed environment, ways in which you can coax them to produce it. And so, yeah, there are lots of different approaches where people are taking to more intentionally identify these types of molecules. But you know, there are many scientists that write lots of proposals to go scuba diving in the Bahamas or you know, other Tahiti or other nice places to isolate organisms that might be interesting from the standpoint of natural products. That still is ongoing. Yeah. Yes.
Okay, so I've heard that there is basically programs to, I guess, take various chemicals and basically just combine them, just organic chemicals to make medicine, and just, well, blasting these medicines, these, I guess, artificial created things, sort of chemicals, and getting like 100,000 of them, and just brute force testing them. And, like, have you done any work like with that, or I guess what do you feel organic molecules to synthesize them into synthetic medicine? I'm probably not using the right terminology.
No.
Like, how do you do that?
Yeah, it's a great question, actually. So the question that was asked was, you know, there are processes where people randomly combine chemicals and they can arrive at, you know, you said hundreds of thousands, but in many cases millions of molecules randomly created, and then just test them for activity and try to identify, you know, active molecules. And so in fact, this happens. This is called a combinatorial synthesis. So you just combine things and you can generate large libraries of compounds and then test them in what is called a high throughput testing, where you essentially build arrays of different either chips or microarrays, and then just test them and see if anything emerges out of that. And so there have been some successes, but I would say that those successes are relatively limited, because often when you combine these molecules in these random ways, you know, a big part of the challenge is to actually isolate out of that millions of molecules the small amount of a particular compound. And so often these things are tested as a mixture of different molecules. And those mixtures can have synergistic effects where they might actually work well as a mixture, but might not work well as individual molecules. And so there are some challenges there. And also I showed to you this sort of landscape of the chemical landscape. And why I didn't show you is that in a combinatorially generated landscape, even if you're combining things in a random way, you're often only accessing a very small part of that landscape. So you don't generate as much diversity as you would expect, even with millions of molecules, as you would arrive at with natural products, which have had billions of years to generate diversity, okay? And so in fact, you access much more of a diverse, more diffuse landscape with natural products than you often do through these combinatorial libraries. And so, but certainly it shouldn't be discounted. That is an important part of arriving at new molecules. And so one thing you can do is start with different natural products that are in different landscapes, and then use that as a way to diversify into other spaces. But this is definitely one of the approaches that, for example, pharmaceutical companies use to try to arrive at new molecules and test for activity. So you have that absolutely right. And what I told you was perhaps the other end of the spectrum, where you're very, very intentionally starting with a particular starting point and looking to advance that because it's already been shown to have some activity that is of interest.
All right, so back of a crab, fungi on the back of a crab is pretty weird. What's the weirdest place that medicine has been found?
Oh, wow. Okay, so I would tell you that all of us are harboring lots of natural products. Perhaps the weirdest place is that in your gut, you have lots of natural products flowing around. So your gut microbiome, this is really emerging. It's a very, very interesting place, okay, where love and wars are fought and everything happens. So turn to your insides if you want inspiration for new natural products. So thank you.
Thanks, everyone. Thank you for coming.